Hello, I'm Camille Dowse, and I did my paper and presentation on the evolution of the finches on the Galapagos Islands. And here are just three of the finches, the medium ground finch, the large ground finch, and the small ground finch, from left to right. And in the background is a picture of the island of Daphne Major. And so first, what is evolution? It was originally defined as a descent with modification, or change in the characteristics of a population over time. Today it's defined as changes in the allele frequencies of a population over time. And so there are 14 different species of finches on the Galapagos Islands, and Darwin was the first to study these birds, but he wasn't the last. Peter and Rosemary Grant have studied them since 1973, and they spend 6 months of every year on the island of Daphne Major to collect data and survey the birds. And so why Daphne Major specifically? Well, it is an ideal study site just because it's super isolated. It is of a fairly small size, and its geographic age is a known quantity. It's a known thing, which is also very important. So evolution happens by many different mechanisms. So scientists have done many studies in order to perform a whole genome resequencing and various other genetic tests. Alright, so first is evolutionary mechanisms, and we have gene flow, introgression, migration, adaptive radiation, speciation, and natural selection for the various evolutionary mechanisms I'm going to talk about today. Alright, so first is gene flow, which is defined as the movement of alleles from one population to another population. And it does result in evolution, but it's usually a non-adaptive evolution. So it can be good or it can be bad. So the 14 different species of finches are in 7 distinct and indistinct lineages, based on genetic monophyly and morphological distinctiveness, both of which are based off of gene flow. Gene flow is highly reduced between the same species of finches that live on different islands because of the distance between those islands. And gene flow is greater on some of the peripheral islands because there isn't as great of a distance. And the benefit of this is just because these islands are more isolated from the same species gene flow, and they also have more extreme environmental conditions. And so it has been documented between Geospiza conirostris on the island of Hispaniola and the Geospiza conirostris on the island of Genovesa by way of the ABBA-BABA test, which is a type of genetic test. It has also been documented for the warbler finch, Curthida fusca, and the common ancestor of the non-warbler finches. So introgression is another mechanism. It's also known as introgressive hybridization. It's the movement of a gene from one species into the gene pool of another by the repeated back crossing of an interspecific hybrid with one of its parent species. It's got generic variation of beak shape has been increased in the finches by introgressive hybridization. Specifically, one gene specifically is called the ALX1 gene, and it was introduced among the different species of finches by introgressive hybridization. The pointed allele of this gene likely contributed to the evolution of more pointed beaks in 1987, after natural selection took place as a result of the change of food supply in the 85 to 86 drought. And so migration is another one. It is the evolutionary definition of migration is the movement of alleles from one population to another. And so the movement of alleles is done by the movement of an animal or a population from one region to another. And so the finches first showed up on the Galapagos Islands by migration, and the subsequent colonization of those islands by their one main ancestral population. And so this happened between 2 and 3 million years ago, as evidenced just by when the islands were formed to their geographic age, and also, or their geological age, and also based off of genome sequencing and genetic testing. And so there's still small migrations today between the various islands of the Galapagos, and there are many of those small migration events in the 90s, even though a lot of those birds didn't stay on the island that they migrated to, their offspring did. And so migrating finches that would come from the Santa Cruz Island and the Santiago Island, they stayed to breed, but then they left after the breeding season was over and their offspring stayed. And so migration plays a key role in the evolution of finch communities. Although the islands of the Galapagos are fairly isolated from both each other and the mainland, there is still only a little bit of migration between the islands. And so adaptive radiation is the production in a short period of time of many species from one occupying different ecological niches. And so there's four main reasons for the relatively quick adaptive radiation of the finches on the Galapagos Islands. And it's first, it's strong isolation from the South American continent. Second, generation of new islands as a result of volcanic activity. Third, climatic changes caused by the El Niño weather phenomenon. And fourth, sea level changes from glacial and interglacial periods, which then resulted in interchanges of both island formation and coalescence. And so it's led to four main groups of finches, when based off of the characters of the bill, plumage, and foraging behavior. And so these four groups are the ground finches, the tree finches, the warbler finch, and the cocos finch. And so modern evidence of adaptive radiation begins with basically the association of beak size and diet. And so there's a very strong relationship between the size of the beak and the maximum size and hardness of the seeds that the finches can crack and eat. And so the finches have different beak sizes, and so they can each fit into a specific ecological niche based on their diet. There's three essential ingredients for adaptive radiation proposed by Darwin: variation, inheritance, and selection. And so phenotypic variation was determined by capture and measurement and just following the offspring for years. Heritable variation in beak and body size was observed by the Grants, Peter and Rosemary Grant, especially in the Geospiza fortis and the Geospiza scandens. And then natural selection is a big thing that helps push adaptive radiation along, and that was observed especially just in the drought of 1977, the El Niño event of 1982 to 83, and then the drought of 1985. Speciation is the process in which new genetically distinct species evolve, usually as a result of genetic isolation from the main population. And so it begins with the divergence of a population, and then it finishes when two divergent populations coexist with little or no interbreeding. There's been 7 major clades determined for these finches using the haplotype network, and then also phylogenetic analyses using mtDNA. And so clades are the result of speciation and are examples of groups of species that have evolved from one ancestral species. One event, the Grants observed on Daphne major, started in 1982 when 2 females and 3 males of the Geospiza magniorostris began to breed. And before this, they hadn't been a breeding species. They hadn't really been established on Daphne major. And so the small population was established by dispersal and colonization, and the beginning of this population was a single pair for the first generation and a brother-sister pair for the second generation. The Grants concluded that speciation starts slowly and with small steps. The medium ground finch and the cactus finch are another example of speciation. They both occupy different ecological niches, but their diets do overlap. And so the two species remain separate because of their beak morphology, which determines what they can and cannot eat, and their song, which is used in courtship displays and breeding. So the very end of speciation is the total absence of gene exchange between species. However, not all of the finches have reached this point. There's 2 lessons that the Grants learned about speciation. It's first that species will diverge in mate preferences before their genetic incompatibilities will evolve. And second, different populations of animals can act and function as biological species even before they would be recognized as species, based only on genetic distinctness. Alright, so natural selection is the last one I'm going to talk about today, and it's the difference between the survival of individuals with a certain phenotype compared with individuals that have a different phenotype. The Grants have determined that it is observable, interpretable, and a repeatable process in a natural environment. It fluctuates in direction, it happens when the environment changes, and it produces an adaptive change which has evolutionary consequences. So they observed 2 major natural selection events on Daphne major. First was a drought that lasted from 76 to 77, which basically left mostly seeds of the tribulus plant, which are very difficult to crack and open. So the finches that had longer, deeper beaks and were large in size were the main survivors. The second event was an El Niño event from the late 1982 to mid-1983, where a grand total of 2,408 millimeters of rain fell, which was 14 times greater than the amounts of rainfall in the previous 5 years. And so the flora of Daphne major changed, and so did the availability of different seeds. And so the finches that had the highest success in gaining their food and the highest survival rate were the finches that were small in size and had small beaks. And so now I'm going to talk about scientific tests and tools to show evolution. First is just genome sequencing, which is figuring out the order of DNA nucleotides that make up an organism's DNA. It has to be done in pieces because all available methods of DNA sequencing are only able to handle small pieces of DNA at a time. So there's two approaches to genome sequencing, and they both involve breaking the genome into smaller pieces, sequencing the DNA, and then putting the pieces back together. And so a group of scientists completed a whole genome resequencing for all 14 of the finches and 2 tanager species that are close relatives of the finches. And so they did a resequencing because one of the species of finches has already had a genome done, and so they redid that one and then included the rest of the species as well. They analyzed patterns of intra- and interspecific diversity, as well as the phylogenetic relationships among the species of finches. And so they determined the phylogeny of the finches. Different phylogenetic splits were able to be dated because of genome divergence. And so the first split between warbler finches and the rest of the finches happened about 900,000 years ago. Rapid radiations of ground and tree finches began about 100,000 to 300,000 years ago. There was evidence for introgression and gene flow, just based off of different genetic tests, the mtDNA, and discrepancies between different phylogenetic trees. And they also found a locus that had a major effect on beak shape. And so this is the ALX1 gene, had 2 alleles with either blunt or pointed beaks. And so the phylogenetic tree showed a profound divergence between the B and P phenotypes that had to have occurred very soon after that first split, 900,000 years ago. The introduction of the pointed allele by introgressive hybridization likely contributed to the evolution of more pointed beaks following natural selection because of the drought from 1985 to 1986. So different genetic tests, they did sequencing analysis to find and show genetic distinctiveness within the finches. The phylogenetic analysis and intron and mitochondrial loci were used to determine the clades of the finches. And then ABBA, BABA tests were used to determine and analyze patterns of gene flow and the total extent of interbreeding in populations of finches. All of these tests were able to show evidence for each of the different evolutionary mechanisms I've talked about today. And they were also helpful in establishing phylogenetic trees and determining when there were different splits and divergences among the species, as will be shown in Figure 1. And then 7 clades were found, which we'll see in Figure 2. So this is Figure 1. Go ahead and pause your video to take a good look at it. And then this is Figure 2. Go ahead and pause your video to take a good look at it. For both of these figures, I will include a description just in the comments of what specifically these are. And so just in conclusion, there have been 14 different species of finches that have evolved over time from one ancestral population. There are many different mechanisms that can achieve evolution, and each of these mechanisms work together to achieve evolution. And the finches in the Galapagos Islands have been studied as examples of evolution, especially natural selection, because they are in an undisturbed habitat and in a very isolated location. And so the easiest place to see the evolution of these finches on the Galapagos Islands is on their beaks. And then genome resequencing, as well as a variety of other genetic tests, have been done to gain more understanding of the evolutionary history of the birds. And these finches, they're not done evolving yet. They're going to keep evolving and keep adapting to their environment. And so an area for further study that would be really interesting would be to use genome sequencing and all the different genetic tests that are available to show the differences in genes and gene expression among the different finches. And then just real quick, here are my references.